Assalamu alaikum dear students. This is your botany sir, and today we are going to continue with the events in sexual reproduction, and today we will deal with the post-fertilization events in sexual reproduction. The post-fertilization events in sexual reproduction begin with the formation of zygote. These events include development in zygote and embryogenesis. In all sexually reproducing organisms, the formation of zygote is universal. That is, zygote is formed in any case, and this is the most important event in the process of sexual reproduction. Zygote is the resultant of gametic fusion, known as the fertilization or syngamy. In organisms with external fertilization, zygote is formed in the external medium, that is, usually water, whereas those exhibiting internal fertilization, the zygote is formed within the body of the female organism. Further, development of zygote depends on the type of life cycle of the organism and environmental conditions. In many fungi and algae, the zygote develops a thick wall and forms a spore called as the zygospore. The wall formed is resistant to desiccation and damage. The wall is formed to overcome the harsh environmental conditions. It undergoes a period of rest before germination. In organisms with haplontic life cycle, you must be knowing, as this has been discussed in your 11th class, the zygote divides by meiosis to form haploid spores, which grow into haploid individuals. In most animals, zygote divides by mitosis to give rise to diploid embryo, and the individual thus formed is diploid. It leads to diplontic life cycle, as in case of the human beings. In most plants, the zygote develops to form the embryo and then the diploid sporophyte. The sporophyte, when mature, produce haploid gametophyte to form the gametes. It leads to a diplohaplontic life cycle, wherein the haploid phase is minimized and brief. Hence, zygote is the vital link that ensures continuity of species between organisms of one generation and the next. Every sexually reproducing organism begins life cycle as a single-celled zygote. Now the next event, that is the embryogenesis. The process of development of embryo from the zygote is called embryogenesis. And during embryogenesis, zygote undergoes cell division and cell differentiation. The cell division increases the number of cells in the developing embryo, and you must remember here that the cell divisions are all mitotic in nature, while cell differentiation helps to form specialized tissues and organs to form an organism. On the basis of development of zygote, animals are grouped into oviparous, viviparous, and ovoviviparous. The oviparous animals lay fertilized or unfertilized eggs. In land animals, reptiles, and birds, the fertilized eggs are covered by hard calcareous shells and laid in a safe place in environment. After a period of incubation, young ones hatch out. In viviparous animals, such as mammals including human beings, the zygote develops into young ones inside the body of the female individual. After attaining a certain stage of growth, the young ones are delivered out of the body of the female organism. Because of proper embryonic care and protection, the chances of survival of young ones is greater in viviparous organisms. And the third, in ovoviviparous animals, the female retains the eggs inside the body after fertilization without providing extra nourishment, as the placenta is absent. That is, there is no connection between the female and the developing this embryo. For example, sharks and rattlesnakes. In flowering plants, the zygote is formed inside the ovule. After fertilization, the sepals, petals, and stamens wither and fall off. The pistil remains attached to the plant. The zygote develops into the embryo, and the ovules develop into the seeds. The ovary develops into the fruit with a thick pericarp, which is protective in function. After dispersal, seeds germinate under favorable conditions and produce new plants. And thanks, that is all for today.